So, very good morning, one and all. Today we are going to discuss about the liposomes. So, already we discussed the two part of liposomes like method of preparations, applications, and advantage, and what are the various types of liposomes. Today we are going to discuss about the liposomes part 3. What are the other method of preparations like the active loading technique. In that active loading technique, the liposomes are first generated containing the transmembrane gradient. For example, the aqueous phase will be inside, the outside the liposomes are different. Okay, both are in the two different way. Subsequently, an amphibolic drug is dissolved in the exterior phase can penetrate the followed with the phospholipid bilayer. So after the permeation interactions with the trapping agents, the core to lock in the drug. Oh, okay. So the inside the aqueous phase, outside the liposomes are different. So in that the antibody drug is dissolved in the exterior aqueous phase can penetrate into the phospholipid bilayer. So when it was established, it is in the year of 1976, and the Damer and Nicholas demonstrated the pH gradient could be utilized to load the catecholamine into liposomes, resulting in stable drug retention in the in vitro. So this is one of the effective loading technique for the preparation of liposomes. When we talk about the stability of the liposomes, generally the liposomes have a poor stability. So we can go for the prevention of the chemical degradation. Start with the freshly purified lipids and freshly distilled solvents. So avoid the procedure which involves high temperature. So we could not go for the high temperature. Then carry out the manufacturing in the absence of oxygen. So oxygen irukkodadu. So deoxygenate aqueous solution with the nitrogen. So we can remove the presence of oxygen with the help of the nitrogen gas. The store the liposome suspension in the inert atmosphere. Include the antioxidant as a component. This is the way we can prevent the chemical degradation. So the stability of liposomes, the prevention of chemical degradation எப்படி prevent பண்ணுவீங்க பார்த்தீங்கன்னா, so annealing, this is the best method to control the physical degradation. Example, incubating the liposomes at the temperature high enough above the phase transition temperature. Okay, the stability of the liposomes may also be increased by cross-linking membrane component covalently by using the glutaraldehyde. Glutaraldehyde here we act as a cross-linking agents, okay, by the fixations. Then osmifications or polymerization of alkane containing phospholipids. Okay, so these methods increase the mechanical strength of the membrane and render them less susceptible to the disruption. Okay, so we can prevent the stability problems by the chemical and physical degradations. Then there are the several characterization, or it is called evaluation of the liposomes. Once the liposomes are successfully manufactured by any one of the technique, it should be carried out by the different evaluation methods. So the physical characterizations, we can go for the entrapment efficiency, the vesicle shape, morphology, lamellarity, the particle size, shape, then surface charge, the phase transition we gave here, the drug release. By the way, the chemical characterizations, we can go for the phospholipids concentration, cholesterol concentration, the phospholipids peroxidation, okay, then pH of the liposomal dispersion, osmolarity. So by the way, we can go for the biological characterization. We can go for the sterility testing. It is one of the important testing. Then pyrogenicity testing, whether it is any pyrogen are present or not. Then animal toxicity test. So finally we can go for the stability testing. So when we see the chemical characterization, there are, as I told, in the phospholipid concentration, cholesterol concentration, the phospholipid concentration that we can go for the Bartlett assay and Stewart assay or by the TLC method. Okay, then pH of the liposomal dispersion that we can normally we can check with the digital pH meter. Then osmolarity by osmometer. Then biological characterization, that we can go for the sterility by aerobic or anaerobic culture. Then pyrogenicity by we can go for the LAL test, limulus amoebocyte lyase test. Then we can go for the animal toxicity, whether is it produced any toxicity on the animals, monitoring the survival rates and histology and pathology of the animals. Okay, finally we can go for the stability of the liposomes. The stability is defined as the capacity of the particular formulation in the specific container or closure systems to remain with the physical, chemical, microbiological and therapeutic and toxicological specifications. That we can go for the any one of the stability method. Okay, the stability testing liposomes, that we can go with the high temperature and low temperature with the minimum period of 1 month, the room temperature for 12 to 24 months, then 2 to 3 freeze the cycles, 25 to 20 to 25 degree, then 60 cycles per minute the reciprocating the shaker for 28 to 24 to 48 hours, then 6 to 8 heat cool cycles is required. Okay, this is the way we can go for the stability testing of the liposomes. Okay, and moreover we can visualize the microscopic examination. So after the storage, the liposomes are evaluated for the vesicle size, shape, is there any changes are occurred in the shapes and size, the number of vesicles are present in the cubic millimeter and the residual drug content, that will be checked. Then characterizations, that we can go for the size and size distribution, we can go for the electron microscopy. This is the most specific method to determine the size of the liposomes. Since it is allow us to view the individual liposomes to obtain the exact information about the profile of the liposomes populations over the whole range of size. Then we can go for the laser lighting scattering method, also very simple to determine the or characterize the particle size, liposome size. Then we can go for the surface charge by zeta potential, the free flow electrophoresis on the cellulose acetate plate in sodium borate buffer pH 8.8. The sample are applied to the plate and the electrophoresis is carried out at 4 degree for 30 minutes. The plate is dried and possible lipids are visualized by the molybdenum blue reagent. The liposomes get bifurcated based on the surface charge. Then we can go for the percentage drug entrapment, then phase behavior. The phase behavior, the liposomes at the transition temperature undergo the reversible phase transition. Example, the polar head groups in the gel state become a disordered to form a liquid crystalline state, which can be determined by the differential scanning calorimetry. Then lamellarity, the average number of the bilayer present in the liposomes can be find out by the freeze electron microscopy. Then there are the several applications of the liposomes in pharmaceutical field. The liposomes act as a drug as a carrier for protein drug delivery vesicles. The liposomes in antimicrobial and antifungal and antiviral therapy. It is also involved in the tumor therapy, gene therapy, immunology. The liposomes as a artificial blood surrogates. The liposomes as a radiopharmaceutical and radiodiagnostic carrier. The liposomes in a cosmetic and dermatology. Okay, so when we talk detailing the liposomes in the gene delivery, the variety of physical and biological methods have been developed for the transferring gene into cells. The most widely used types of vesicle for gene delivery are the viral and non-viral technique. So non-viral vector systems, the liposomes and lipid complex, especially engineered liposomes such as the pH sensitive liposomes, cationic liposomes, fusogenic liposomes, genosomes, lipoplex and lipopolyplex have been extensively investigated for the gene delivery for the potential. Okay. Then the liposomes also act as a drug delivery carrier for the several drugs. There are several methods, several modes of drug delivery application have been proposed for the liposomal systems, where some of the pathogen caused disease like the enhanced drug solubility like amphotericin B. Okay, so amphotericin B, the production of sensitive drugs molecules like DNA, RNA and ribosomes, enhanced intracellular uptake. Okay, like example of anti-cancer drug and antiviral and antimicrobial drugs. Okay, The liposomes, it is also involved in the tumor therapy. The most of the medical applications of the liposome have reached the preclinical and clinical stages in the cancer treatment. It have been demonstrated that all small and stable liposome can passively target the several different tumor because they can circulate for the prolonged time. When the drug will be present in the body with a prolonged time, it will be very effective. The targeting the liposomes can be used in the different way. The natural targeting of liposomes, the use of long circulatory, use of ligand mediated targeting. The liposomes are also involved in the cosmetology and dermatology. In early studies, the liposomes containing the stratum corneum lipids have been tested in order to enable the better skin penetrations. Okay, the liposomes, it can be penetrated by the skin permeation. The two mechanisms for enhancing the topical localization are targeting of the liposomes and encapsulated bioactive. The transepidermal pathway, that is a skin layers, the transfollicular pathway, the hair follicles and glands. The liposome based anti-aging formulations are now well developed, like in the form of creams, lotions, gels and hydrogels have been formulated and launched in the cosmetics market in the year of 1986. The L'Oreal Pharma company. The liposome preparation reduced the roughness because of its interaction with the intracellular lipids, resulting in the skin softening and skin smoothing. So there are the several marketed products are available in the liposome based drug delivery, like the liposomal lamboterizine B. It is used for the treatment of fungal infection. Then it may be available in the form of the different trade name, the like the ambisome, the fungal and protozoal infections, and cytarabine. It is also malignant hypobome meningitis for the cancer treatment, like tanurubicin. It is also HIV related sarcoma. There are several liposomal IRV vaccines that are also used for the treatment of hepatitis A. The liposomal IRIV vaccines for the influenza. Okay, likewise there are several liposomes formulated. The product may be used for the different category of disease, especially for the gene drug delivery, then cancer treatment, then cosmetic and dermatology way. Okay, the liposomes PG toxorubicins, it is also used for the HIV related capsoma sarcoma and used for the blood cancer and metastatic ovarian cancer. Okay, so there are the several marketed products that day by day increased in the form of liposomes. It is very effective to treat the patients. So thank you very much for patient listening. Hope you understand the liposomes, the characterizations and applications which are widely used for the pharmaceutical preparation. Thank you.